Good evening. This is Fred Loxam, and I'm a representative of the San Antonio Native Plant Society, and I'm thrilled to have O.W. Van Auken, or Bill Van Auken, be with us this evening to talk about a very interesting topic. Bill is Emeritus Professor at the University of Texas at San Antonio, and he's in the Department of Integrative Biology, which is a really good department, really good kind of department to have. Bill has been doing some excellent work, and he's going to describe it this evening. So, Bill, I'm handing the program to you.
Thank you very much. I'm interested in, well, I'm a plant biologist, plant ecologist. I'm interested in maples for sure, but other species here in Central Texas as well. And I'm going to try to talk, I want to talk, and I'm going to talk a little bit about maples in this presentation. And let me see if I can do this correctly. Now, plant communities of the past, of Texas past, are difficult to interpret. And the reason being is that most people didn't write anything down, or if they did write it down, it was expunged from the literature. Some of these things are in Spanish and they are in archives in Spain, but most people don't have access to them. And so it's one of those things, you have to do some interpretation, and there have been so many changes in Texas, partially because it's a private property state, but there have just been changes going on since the very earliest times, and they're difficult to interpret. So because of that, the populations that we see, or the communities that we see in the present time, are difficult to interpret as well. And the future, well, futures are difficult to interpret for sure. I want to start with something that's interesting to me, but Niels Bohr is a Danish, he's passed away, a Danish physicist. And I guess that's interesting, especially since our host is a physicist. But what he said was prediction is very difficult, especially about the future. And I agree with him. And you can keep that in mind as I go. And if you have questions, we can talk about them, or you can send me a note or send Fred a note and he can get in touch with me. But most of this stuff that I'm going to talk about is in fact published, and it's available online, so I can give you a reference, or Fred can. But let's go ahead and get started. Now, global changes are going on. I'm sure all of you understand that. But are all of the modifications we see to the environment due to global changes that are going on right now? No. And are they direct changes or indirect? Well, the answers are really no and no, but you even have to take a little bit of a grain of salt with that. Local conditions are more important in many cases. Global change effects are hard to detect in some cases, in many cases, but they are occurring, and I believe that. But many of the things that I've been seeing are due to conditions that are nearby or close. And it's hard to deal with these global change problems. Our climate, for one, has been changing forever. And it's been getting warmer for the past 20,000 years, approximately 20,000 years. And we're supposed to be getting into a cooler climate, but that doesn't seem to be happening because of things that we have been doing and are continuing to do. But anyway, let me talk about some some things related to this maple. What I'm going to do is to show you where these populations are generally found. We're going to talk about the plant a little bit, its characteristics, then the light level that the plant responds to, including photosynthetic rates. And then we're going to look at some growth, survival, and recruitment of maples into certain populations. Where does it occur? Well, it ranges, as you can see here, I think, in Texas, in West Texas, and then up in New Mexico, various places, Nevada, and then, of course, Arizona. And then there are lots of populations in Utah. There are even populations in Western Oklahoma. And then there are a couple here that are not shown in this particular figure, up near Waco in some of the canyons there. Texas stands are fairly isolated. In fact, there are a few stands in Mexico too, but they're in higher elevations. But so are these out in West Texas. This is the Big Bend area. These are the Davis Mountains here. And then up here, these are the Guadalupe Mountains. And all of these areas are very interesting to visit botanically. And of course, there are lots of interesting animals there. And this big national park here was established because of its geology. So anyway, there are a lot of interesting things to study about the maple and its associated communities where it occurs. Now, this bigtooth maple, it's called Acer. Acer is the genus, and then grandidentatum is the species. It was once considered a subspecies of sugar maple, Acer saccharium, which is a species that is very common in the eastern United States. Some people can still consider it a subspecies. And it's the family at Aceraceae or Sapondaceae. Again, depends on who you read and who you're looking at. Aceraceae is fine with me. This is a very nice picture of one here in Central Texas. It's a small to medium size deciduous hardwood tree, hardwood maple. It's drought resistant and has different forms that you can find depending on where you are. Rocky slopes, usually multiple stems. They're not nearly as big as those you find in the canyon bottoms that may have one stem and be up to 15 meters tall. This is another picture. It's one of my favorites. That's why I'm showing you. But this is a major tree that we found out in well, just west of Bandera in the ABK Preserve area. You may not know about this ABK. Its property. It's almost 5,000 acres that was donated to the state of Texas to be a natural area. Well, this was over 10 years ago, and it's still not accessible to the public. This is me here, and this is Xinjian Chen. Well, she was a former student, but a colleague, and she helped with some of these projects. And what we're going to do is measure the basal diameter of this particular bigtooth maple tree. And that's what we did in a lot of places, getting some ecological information about the species. Now, if you're interested in the future of these maples, you don't usually look up to see them in the canopy, but you look down to see what's in the understory. These are some juveniles here. Once they get past the first year and drop their first leaves, it's hard to tell how old they are, but they're juveniles for sure. They have opposite simple leaves, and here are some examples here. And they have these big indentations, and that's the reason they call them bigtooth maple, but there's a huge amount of variability. These are the female flowers, and I say here, the dioecious yellow flowers. Well, the trees aren't dioecious. That is, they're not separate sex trees like the juniper that we have here, but they have male flowers and then female flowers in the same plant. These are the females here, these were the females, the ovaries, and these are the seeds or the fruits. They're called double-winged samaras. Now, we were interested in why these juveniles couldn't get to the canopy to become trees. So we were interested in plant mortality. And this is a relatively early study, but it's a pretty small one. But we had 6 plants, we protected them, put them in cages, and the protected ones survived. Survival after a year was 33%. We had 2 survived for the first year. But the plants in the open, no cage, they're unprotected. After 1 year, the survival was 0. So what happened to them? Well, cages keep the herbivores away, and they keep the plant protected. And so we have a major feeling that the herbivores that are in these areas are a main cause for the juveniles not being recruited into the adult population. Here's a picture of one of the cages we use. This is wire, well, all this stuff we bought at the hardware store. Metal rods here, reinforcing rods, and then plants down here. Now, we have the plants surrounded, but we also have tops on these cages. So rodents, including squirrels, can't climb over the top and get in and consume the maple. That's one of the things that we found fairly early on, and we did, and I'll come back to that some more shortly. But we looked at the importance of different light levels on the growth of these plants. Here's another example of the leaves, and you can see these big insertions here that sort of define this particular species. But if you look at seedling mortality, and these are seedlings, and you look at it over a range of light levels from, well, dark would be way over here. This is 20% of ambient, 40, 60, and 100%. At full light, in this particular experiment, we had 100% mortality. So this is ambient light, but then mortality is 100%. And these stars indicate significant differences. At 60% ambient light, we had about 20% mortality, but at 40%, this is shady conditions, there were no mortalities. But then if we make it even darker, 20% of normal light conditions, we get about 40% mortality. So they like it in shady conditions. That's where the juveniles usually survive if the herbivores don't get them. Now, this is how the leaves respond. These are sun leaves. These are at the top of the maple canopy. And these are shade leaves. So sun leaves up here, shade leaves down here. And what this is a light response curve. So we made this with a photosynthetic meter. This is full light here, and then this is the 0 over here. And then this is the mean photosynthetic rate, micromoles of CO2 taken up per square meter per second. If you do the ANOVA, there's no significant difference between all of these measurements for the sun leaves versus all of these measurements for the leaves in the shade. So these A's up here mean that this one is not, and this is another statistical measurement here. This is one of the multiple range tests, but the A's suggest significant, there's no significance between this population or this light level, et cetera here. And you start to see some differences here finally. And then when you go to this level, and the light level here is about 200. So it's very low. It's 10% of the maximum over here. But you start seeing differences here. And then you see some down here too. But most of these in the low light conditions are not significant. So these are light levels. This is photosynthetic rate. Overall, no significant differences. And when you look at the individual values, you find that they start to diverge down here. And you can see that this is where they start to reach a plateau. Now, the same thing is true down here. But if you go a little farther, and you're not really supposed to do this, but if you compare the values here versus the values here, you find that there are some significant differences in some of these, but you're really not supposed to do that. Anyway, we had to look, and that's the kind of thing we found. So for the most part, the sun leaves are not significantly different from the shade leaves, although the values that you see here are a little bit lower. And these photosynthetic rates here are lower than most of the plants that you see in the full light. For instance, if you look at some of the C4 grasses, I mean, they are 6 times higher than these values. So these values are fairly low, and they're lower still when they're in this deep shade down here. Okay, now we're going to go out to this ABK Preserve, and we're going to look at some other things. This is the Albert and Bessie Kronkowski, State Natural Area, and it's about 5 miles west of Bernie. Hey, thank you. If you go out to Bernie and then head west towards Bandera on Highway 46, about 5 miles, and you'll pass it. And, you know, you'll see the fence, and the fence has a couple of gates, but they're usually locked. But this is where we've done some of our studies. And the kind of studies we did here was to see what the populations of the various species were in the communities that we could tell were bigtooth maple communities. Now, let me show you what we did. So we rented it. Well, we got a friend of one of my co-collaborators to fly us over this area. This is the airport out in that direction, out towards Bernie. And he took us up, flew us around over the preserve. And this is what you see. Well, this picture was taken by my colleague, Donna Taylor. I was in the front seat I was in the back seat next to the pilot, or behind the pilot. But by the time I finished getting my camera adjusted, we were in the next county, you know, so Donna took the pictures pretty quick. But anyway, this is the ridge. This is a ridge here, and this is a hilltop. And then this is a canyon. And these pictures were taken in the fall of the year. And these deciduous plants, the maples, but not just the maples and the black cherry, the black walnut, and a few others will change colors. So we can tell that these plants down here in the bottom of the canyon are deciduous plants, but we can't tell what kind they are. A lot of these up here are certainly juniper, and some of these other ones are live oak. But down in the canyon, these are bigtooth maple. Now, not in every canyon, but in a lot of them in this area. And sometimes you find just a few, but in this particular canyon, we found quite a few. So what next? Well, here's another picture. This is probably a maple here. This may be a cherry or a cedar elm. They occur down here too. But you can see that these are definitely different than these up here. And this is the hillside, whereas this is the center of the canyon. Now, down on the ground, you get a different perspective. You figure, oh my gosh, where are these maples? Well, this is just looking out across this area, and you can see juniper, live oak, and a few other species, but it's pretty thick. Well, we headed down the hill, and you can see it's also steep. Here are some of the maples. This is where we want to get to, but we have to go this way through all of this brush. And on the hillside, you can see how steep it is here. These trees are growing pretty much straight up, and this is the slope. So when you're climbing down, you got to be ready to, sit down and slide a ways on your rear if you don't want to go tumbling all the way down to the bottom. But this is the kind of situation you see in this area. This is down in the bottom, and this is the drainage here. Of course, it's dry most of the year, and a lot of these trees here are maples, and this is myself, and I think this is either Donna Taylor or Chin Chin here. But what we did is we surveyed the populations here to get an estimate of how many of these maples we had. And we also wanted to look for juveniles as well, because we were going to cage them. And I'll show you some more of that information shortly. Well, what did we find? We found, and this is an aerial view that we took from the plane, that there are several populations that we studied here. And we probably could have run another transect line up here, but these are transects that we measured on the ground, looking at the various plant species that were here, woody plants for sure. And what we found is that the bigtooth maple had a density, this is measured on the ground, of about 800 plants per hectare. And then you have juniper, and this is black cherry here, and then we have a number of other species. But in these communities that we're looking at, the relative density was about 800, and this is about 52% of the density in this whole area. The total density was about 1,300 plants per hectare, but a lot of variability, and you can see that this is about half of it. The juniper about 25%, and all the rest of the individuals are they make up the final 25%. And then knowing this and knowing the area of these communities, we projected how many we could find in the whole community. And so it looks like they're quite a bit. And you think, well, maybe they are recruiting, we just haven't seen them. So what we decided, first of all, we aged the maples. And here are some cuttings of the trees. And if you say, you went out and cut all these trees down? No, we didn't do that. These were lying on the ground. These were dead. And this one has heart rot here, and which is true for most of the trees in this area. So they get heart rot, and then beetles get in here and feed on them, and all kinds of other things happen. So it's pretty tough to get ages on these, especially for the real tiny ones. But you can see here, these are annual rings here. So this is the early wood where you have the big cells and they're light. And then this is the late wood where they stop growing or they grow slower and slower until they quit. So this is 1 year's growth or an annual ring. So you've got one here and then you've got others in various ways. But this is a tedious process, but we were able to do it. And we have a pretty good idea of what the growth rate is, at least for some of the individuals in the population. And when you do that, what you can do next is a distribution. So what is this? Well, these are the 1, 2, 3, 4, 5 communities that we studied. And this is a summary down here. Well, what are we looking at? This is the count, the number of trees that we measured. And then down here, this is the diameter. But in addition, since we know how old some of these trees are, we could age them. And we found that some of these started in the 1700s, the very early 1700s. And our experiment was over in 2016. So that's right down here. But what these are estimates of what the population looks like. And the N up here, this is the number of individuals that we measured in this population, the same here. And we summed them up. So we looked at, or we measured 257 separate trees. And there are a couple of interesting things. You see, there's one individual way out here. Well, that started in approximately 1701 or 1700. That's 300 years ago. So these trees, they can last a long time. But as you can see, not very many last that long. But the other thing that we were interested in was, what about these individuals down here, the juveniles that are entering this population? Well, if this population is going to be sustained, you have to have juveniles entering the population. We only found one juvenile in these 5 communities that we looked at. So the zeros here, now these are not the individual seedlings, but these are small trees that are supposed to be recruited into this overall population. Well, the mathematical way you show this is that this is declining here, coming this way, or there are no individuals entering, or very few entering this population. And then they die off over here. Well, so this population looks like it's fairly stable. And you look at the numbers, and there seem to be quite a few in this mid area. And of course, that's why you get this kind of a distribution. Well, this is from approximately 2000 back to about 1970. So there was something interesting going on here that was maintaining this population that certainly stopped at this somewhere down here. What that is, well, that's why I have trouble predicting the future. I don't know for sure, but I'll tell you one thing I want to look at is the effect of herbivory on the individuals that are way down here in this population. So let's continue. We had 2 populations in 2 different canyons. One was planted. That is, we raised the seedlings. They were in the first year, and then we took them out in the field and planted them in areas where there were adult male populations. There were adult maple populations, excuse me. And then we looked at their mortality or survival in time. And that's what I want to show you a little bit more about. So this is a brand new seedling. How do we know? Well, if the cotyledons are still attached, then it's in its first year. The cotyledons are the seed leaves or the first leaves. And we had 2 populations that we worked with in 2 different canyons. But in the 2 different canyons, we had individuals that were left in the open, like this one. And we had others that we caged or protected, like this one. So we had 25 in cages and then 25 in the open. And there were other maples in the area, maple seedlings in the area, but we weren't looking at those. But we hypothesized that the large herbivores in this area, in Central Texas, but in this canyon for sure, were consuming the juveniles of this male population. Consequently, we weren't getting as many recruited into the population to sustain the population. This is another example here, and then we had them flagged, and so we could find them. And they were through this whole canyon. Well, what we found, so I think this is the planted population, but both the planted and the native population responded the same way. So if the juveniles are in enclosures, you have one mortality in 25 over the first year of the study. This is sort of the end of the growing season here. But if they're in the open, you can see that there is mortality to start with, and you lose 6 plants pretty early on, and then the population seems to stabilize until the end. So this is the kind of thing we're looking at. We have carried this population, these populations, and this experiment for almost 4 years out to here, and it's still ongoing. And what you can see, if the plants are protected, you end up with, this is the first year, and then one more mortality in the next year. But the largest percent of the population remains alive. And this is in the planted or the native population. For the open, that is the plants in the open who who the herbivores have access to, you find there are additional mortalities, and 10 out of 25 are gone in the first by the end of the 4th year out here. We did something else. We looked at survival, and of course that's just basically the reciprocal. But we looked at percentages here. So this is 100% when we started, and then we look at the number that the decline or the number of mortalities over this whole time period. And this is just a simple plot. But what I want to do is some regressions to see if we can predict how long this population that are in the open will survive, that is somewhere out here. And the same with these that are in the enclosures. And what we saw was that if you do just a simple regression, a linear regression, and these don't always work, and these plants may not be responding in a linear way over their entire lifetime. But here, up to 4 years, this is looking at those in the open, and this is a linear regression. The R squared, this just tells you how good a fit you have. It's 0.80 or 80%. But the ones in the exclosure, the R squared isn't as good. You only have 50%, but you don't have too much variability here in this population, and only two mortalities, or I think it's 98% survived here. All right, let's look at some other thing. So what I wanted to do was look at the expected population extinction time. That is the time to 100% mortality. And then I wanted to predict that from these simple linear regressions. So each experimental treatment was looked at, and we projected or looked at the data going to 0 with various types of equations. For the exclosures, or with the exclosures, the extinction, that is when you expect to get all of these individuals dying or leaving the population is 60.5 years. Well, in 60 years or 61 years, you would expect some of these individuals would make it to or be recruited into the adult population. Now, if you don't protect them, and this is the same data that I was showing you before, the extinction happens in about 12 years. That is, they go from 25 live to 0 live in 11.9 or 12 years. So that seems to be the reason that you don't have as much recruitment. You're losing the population, and they seem to be lost because they're not protected from the herbivores in the area. Now, the study is still going on, but we've been doing some other things, and I want to tell you about those, and then we'll come back and review the whole thing briefly. But Texas Parks and Wildlife wanted us to do another project, and they want us to look at the maple or the deciduous communities in other canyons in this area. Well, of course, if you have tried to get onto private property in this area, you know it's difficult. people don't want you there studying their plants and animals. And especially they don't want you if you're related to some government organization. Well, the University of Texas is sort of a government organization, and so is Texas Parks and Wildlife is too. But we were able to get permission to go on to a number of properties, and we were going to, what we wanted to do is find the location of new deciduous woodlands that nobody's looked at in the past, and look at bigtooth maple populations. But this is on, again, on private property. So what did we do? Well, we started with aerial photographs, and you look at this and think, oh my gosh, I can't see any specific plants in this area. But what I want to do is take you to a point right up here, because we looked at these fairly carefully, and we looked at a bunch of them. This is out in, I don't know whether it's Medina or Bandera County, but in that area, out towards Lost Maples. And, you know, you can see the roads here pretty clearly. And I want you to look at this one. This is a dirt road right here, but notice the loop. That's distinctive. And then from this, we're going to look at this point over here. Okay, now closer up, you can see, well, there's a lot of color here. Well, this picture was taken in the fall. This is from the satellite. And believe it or not, you can tell that there is a deciduous community here. This is that loop I was showing you. So we're in the exact same area. And in other drainages or dry creeks, you can see there is some color, some change, and a suggestion of deciduous communities, deciduous plant communities. Well, we want to look at what we thought was the best, so we took this as one of them. And when you look closer, you see, and this is not the best photograph, but it's the way it turns out. These are all plants that either have lost their leaves or in the process. The picture was taken in the fall, so you can see the leaf color here. And this is the area where we were doing a survey on this particular property. You can also see that, well, these are obviously not maples here. And these other ones, it's hard to tell for sure. But we did ground surveys on these different communities. Here's another one. Again, you can see this looks like it's a deciduous community. This is probably a dry stream over here. Now, what are some of the general characteristics? Well, I'm going to kind of rush through this, but I still want to show you that we have some basis for our data. We selected 8 different properties from the aerial photographs that looked like they had areas of deciduous communities. This is the area of this one, and you can see about almost a hectare. Here, this one's 4 hectares. This is 1 hectare. This is almost 1. So that's what we were looking at. And we looked at the number of species in the overstory. Again, this is on the ground with, you know, fairly typical ecological measurements, quadrats. But we had usually 5 or 6 species in the overstory. Here, there are 14. But we found a total of 17 different species in the overstory. For the understory, more species, and these are just woody species. And you can see the numbers here, and there's some variability for sure, but we found a total of 30 species in the understories in the conglomerate of these different communities. Overstory density, well, you can see there's a huge amount of variability, and this is the reason for those large plus or minus signs. 183 plants per hectare here, and 1,024 here. Well, the mean value is about 559. So that's the density, the mean density that we found for the overstory plants, and that's plus or minus a bunch. For the understory, again, you see the same kind of variation. 17,000 plants per hectare here, and then whereas down here, almost 4,000. So a lot of variation. The mean, 7,963. And then the basal area about 25. But again, a lot of variation. All right, let's look a little farther. What are the species? Well, we have, again, a bunch of them, and this is not a list of all of them, but this is occurrence. That means that this species, this is the juniper, it occurred in every community we looked at. In addition, the maple occurred in every community, but the others were scattered at various levels. The mean density here, 221, 169. But compare this one. This is one I want you to compare. The relative density of the juniper was high at 40%, and the maple was fairly high, 30%, but the maple was less than the juniper. That's the number of individuals that we found. But if you look at the size, look at the maple. The maples are pretty big compared to the juniper. And it's 32% relative basal area for the maples. So we felt like we were looking in the right deciduous communities to see these maples. For the understory, curiously, Celtis, this is hackberry, occurred in every community, but we didn't have any adult trees. I think we found one in the whole study. And then several maples, oaks here, red oak and lacy oak, and then chinkapin oak down here. So the oaks were pretty important. Mean density was tremendously variable in the understory as well. And here the standard deviation 2,000. I mean that's compared to the mean right here. So a lot of variability. Or if you want to compare them directly, and this is better. Now this is a log, but this is the overstory, and then this is the understory. So a number of species we found, 7 here, 16 over here. Density was lower in the overstory compared to the understory, and then the basal area we only had a measurement for the overstory. But these are log values, and that's the only way you could put all these things in one figure. These values are so low, and these, of course, are so high. That's the reason for it. Okay, how about the distribution? Well, this is the bigtooth maple. So we found a couple, well, 4 smaller individuals here, but not many. And this is over the whole area that we studied. And then we had one that was in this last size class over here. So one old one. But the majority of them are right here, like in the previous study that I showed you. Now, this is a linear, well, actually, I mean, it's a straight line through the population. But you can see that's not a very good descriptor of this population, and that's true. This is a quadratic one here, and it better fits. It's a very good fit for the data, but it's not statistical. It just shows you that that is what the data looks like. If you look at the linear equations, I mean the r squared, 0.4, with a second order quadratic, it's a better fit. But the best fit is here with a 4th order quadratic. What that means is you have 4 terms, x to the 4th, to the 3rd, to the 2nd, and then 1x here. So if you treat the data that you have with this equation, you get a very good fit, 90% fit. But that's not a prediction. Now, what are the overall findings? Well, in Central Texas, these maple populations are found in relatively steep limestone canyons, and in some cases on north-facing hills, on hill slopes mostly. So both in the 2 places you find them, but this one is you don't have very high populations here. The factors that determine recruitment are not well understood. Photosynthetic rates are not different, so they carry out photosynthesis fairly well in the shade, and there's no difference between the two of them. But there's not enough juvenile recruitment to sustain mature populations if they're not protected. So if you have almost zero recruitment with the current native white-tailed deer populations, and that's approximately 1 deer per 5 hectares. That's what you find in the hill country in central Texas. And that's probably the highest deer population anywhere in the country. So it's high, and they will eat maples, although they don't in other areas. The deer don't like them in other areas. Well, if you don't have anything else to eat, guess what? You take the best or whatever you can find. With a reduced deer population, and this is what we would find at the ABK Preserve, because they have a high fence, and they hunted until they didn't have very many deer. So you can see what the number is here. Well, this is, and when you have a situation like that, the adult population unprotected will go to 0 in about 12 years. But with no herbivory, no large herbivores, no white-tailed deer, this is in the protected juvenile Acer population. They are expected to go to 0. Again, this is with a linear projection in 60 years. So in that time period, you'd think that some of them would get out of the zone where the deer can eat them and be recruited into the adult population. So it should be enough time to reach above the point where herbivory is a problem for them. Unfortunately, the importance of the carnivores, and these would be the mountain lions and the black bears, in Acer or 2 Acer populations, and probably other deciduous tree recruitment, it's either unappreciated here in Texas, or it's not believed or not accepted. And Parks and Wildlife, they set the hunting rules, you know. So you think, well, they set the hunting rules so that the deer populations would not be so high that the plants would be in jeopardy of becoming extinct. But that hasn't happened. Well, anyway, are the causes of Acer population declines proximal or distant or distal? Well, that means close or far. Well, they're close. I mean, certainly the climate things and the lack of rainfall this year is going to be a problem to them. But more important, I think, and based on our data, is that there is not enough predation or there are not enough predators to allow the recruitment of individuals into the adult population. Okay, well, keep in mind that this is projection into the future, and you always have to say, well, maybe this is true data. It's positive, and we're following the population, and this will be our 5th year, and next year the 6th year. So I should be able to tell you more in the future, but it doesn't look like the populations, if they're unprotected, are going to be recruited into the juvenile populations are going to be recruited into the adult population unless they're protected. Okay, a lot of people helped. You can look through this, but we all got bumps, bruises, poison ivy, scratches, and I appreciate the help all these people gave, and they're still giving, or at least some of them are. With that, I would say thank you very much. And do you have some questions that you want me to try to answer?